Anyone? Have you all gone through all the lecture notes I've given? Yours? Okay, so today's lecture is going to be really short, and then we will have discussion and see what have your plans for your group projects, and then so that we know that all of you all are in the right direction and not having overlapping projects. So today's lecture is going to be on databases and tools in comparative economics. Before I move on, maybe I want to ask a question. What do you all understand as database? Can you give examples of database? NCBI? Gene bank? Okay. Right, good. So you sort of have idea what a database is. What about tools? Is it different from database? Can you give examples?
Mega form.
Sorry?
Mega form. Call mega form.
Mega. Okay.
Oh yeah.
So what are tools? How would you
That's
define
it.
tools?
Juga.
Tools used to help isolate information to simplify us to read it better.
Okay, good. So databases are basically collection of data, right, in an organized manner. And one very famous one that you know of is Gene Bank. Gene Bank has collection of nucleotide sequences. And tools, you used a lot of tools already in your previous semester of bioinformatics. If you remember, you use tools to analyze, to interpret, to improve things, to work on your data. Right? So today's lecture is going to be about that. Firstly, what's data and what's database? Data is anything that is raw and unorganized, uninterpreted facts, right? And in your case, you're learning bioinformatics, comparative genomics. You are only looking at nucleotide sequences or amino acid sequences. So these are the data that you're referring to. So it could be in any format. It could be FASTA, could be FASTQ. There's all these names. Jog your memory. You all know what's FASTA? FASTQ? Question? Do you know what they are?
Yes.
Which are?
File type.
File type. Okay, they are formats of nucleotide or sequences. Okay, so those are data which you just have. You just have sequences that have no meaning unless you annotate them, you assemble them. You don't really know what they represent. It's just sequences, A, B's, and B's, and C's. Okay, while databases is a collection of data in an organized manner. So you have, for instance, your gene bank. When you search a name, a gene name, or a species, let's say you're studying E. coli, you search for E. coli genes or E. coli proteins, you will get an entire list of genes or proteins related to E. coli. So that's a database that archives data from your experiments. So, of course, there are many different kinds of databases, but we're only going to be looking at biological databases for your course. Examples of biological databases include PDB. I think you also are learning this in your molecular modeling course. Have you all gone through PDB yet? No? Okay. So Protein Data Bank is one of the databases you'll be learning there. Okay. And then there are many others. Have you heard of UniProt, SwissProt, Interpro? Any of these names yet? Okay. You will be learning some of them in your molecular modeling as well. Okay. And so these are examples of biological databases that has collection of biological data. And this data can be of a specific nature, so it could be just nucleotide data, For instance, if you look at gene bank, you have nucleotide data. If it some only protein data, so if you look at protein data bank, you only have experimental data for proteins there. And then there are other databases which are species specific. Okay, so there's something called FlyBase. This is a database that only has genome annotations and protein sequences of fly, the model organism. So as such, there are many different databases you will get familiar with in this course. Okay? Of course, you're not going to be using them all, but you just need to know that there are databases available. So if you are not, if you don't have the funds to start a project from scratch, that means experimentally doing something, sequencing and generating data, you can always rely on these databases to get data from previously, people who worked on it previously already. So you have genome data of organisms, model organisms, transcriptome data as well, right? So databases first emerged as a response to huge data generated by low-cost sequencing. So by low-cost sequencing technology, here you're looking at Sanger. So per sequencing reaction is about 25 ringgit. So you're looking at sequencing a gene, just one gene, not my whole genome. Okay, so you can get it for 25 ringgit, for instance. So it was that cheap. So everyone started doing gene sequencing. Okay, and so they wanted a place to store all these data that people have been sequencing. That's how databases started. Okay? So that's how NCBI and GeneBank started. So one of the first databases to emerge was the GeneBank, which has a collection of all available protein and DNA sequences, and is maintained by NIH and NCBI. So this is in the US, and of course they have their Japanese counterpart and their European counterpart as well. Now, the next term I would like you all to know about is primary database. Okay, so databases can be divided into two, primary and secondary database. So primary database is where you have sort of like raw data that you generate from your experiments. Okay? So it's populated with experimentally derived data such as the nucleotide sequence, amino acid sequence, and even macromolecule structure. So for instance, in Protein Data Bank, PDB, that database has information on X-ray crystallography structure, NMR data, and all that. Okay? Experimental results are submitted directly into this database by researchers, and the data is essentially archival in nature, meaning 20-30 years down the road, if you've lost your original raw data, you've lost your hard disk or hard disk crash, if you have uploaded in this database, you will always be able to access, you'll be able to download it again. So your juniors or anyone else 50 years down the road who wants to pick up from where you left, they will be able to, because this database is being maintained. Okay? And then once a database, a session number is given, the data is never changed, unless of course the submitter themselves write in to say that they want something modified or updated. You can write in, but it takes time for the NCBI staff or the staff of the database to get back to you and they update, right? But they don't delete. At least for now, Trump hasn't done that yet, so your data is still safe. Okay? Right. So as I said, there are three counterparts. We have NCBI, which is hosted in the US, and the same data can be found in DNA Data Bank of Japan and in EBI or EMBL. So all three databases have the same information. So if you have uploaded into NCBI, in a few days you will notice that it will be updated to EBJ and EBI as well. The same the same accession number will be there. Right? And if you look at these features of these nucleotide databases, it's also a primary database, meaning once you get a sequencing data, you can upload as such in your FASTA format or in your FASTA format. You don't have to information about the submitter, that means your name, your email address, where you got the sample from. So let's say you got an E. coli strain from the garden, okay? And then you sequenced it, and then you want to upload in NCBI. So you need to give the name of the strain. You need to give the name. You need to give the GPS coordinate where you collected the E. coli from. How you, sometimes they even ask how you culture them, so what culture media was used. All this information will be required when you're submitting your data. So that's considered metadata. Are you all able to understand? You want to follow? Okay. So that is provided as like a supplementary to basically giving you information about the sample that you're submitting. Right, so they accept any nucleotide sequence submission, and they exchange new and updated data on a daily basis between them. So once you upload in one of these, you will notice that by the very next day, the other database also has it already. Okay, and they house original sequence data, so there is no, they don't modify the data that you upload, they provide as such. Okay, and these three databases, they make up something called IMSD, which is International Nucleotide Sequence Database, through exchanging of data and information between them. So they have meetings very often between these three staff of these three databases to ensure that things are up to date. Right? And aside from that, they collaborate with Sequence Read Archive or SRA. So that is another member database that archives raw reads from high throughput sequencing. Right. So the original gene bank will only upload gene sequences, so single gene from a bacteria or from a human. But if you are uploading raw data from your Oxford nanopore sequencing or Illumina sequencing, let's say you have sequenced the whole genome of the bacteria, then you need to upload in a place called SRA, Sequence Read Archive. Right? They have SRA in NCBI, they have SRA in EBI as well, and in DDBJ. So once you upload, let's say in NCBI, the same data is also available in the other two databases. Okay? So remember that when you look, when you're eventually now you're going to start your project already, right? When you look for genome sequences, you cannot look for it in gene bank because gene bank only have single gene sequences. You need to look for it in SRA or Sequence Read Archive. That's why they have the whole genome. Okay? Okay, the other database or the second classification is known as secondary database. So this comprises of data derived from results of analyzing your primary data. So your primary data is only a gene name, just a sequence name. So you don't really know what they do. Annotations are not normally there. Right? So when you analyze the primary data is when you get secondary data. So let's say you get your original genome sequence from Sequence Read Archive from SRA. Let's say it's an E. coli genome. All you get is multiple. If you recall last semester, you get your raw data from EBI, right? And they are in FASTQ format. If you remember, okay. So that is your raw data. Once you go into secondary database, what you get will be annotations of genes and protein functions. It's no longer just genome sequence. Right. So they are often drawn upon information from numerous sources, including other databases as well. And they are highly curated. That means people have manually or using some algorithms, they have already analyzed this primary data and uploaded this secondary data into these databases. Okay? So it's usually a complex combination of computational algorithms, manual analysis, and interpretation to derive this new knowledge from your raw data or your primary data. So it usually, if you look at secondary databases, you normally see conserved sequences, active site residues, signature sequences, protein families, RNA families, protein domains. Those are the kind of information from secondary database. So it's already more refined. It's no longer just biological meaning to the information. Right. So these are some examples of secondary DNA databases. You have your Interpro, your protein families, which have a lot of member databases now. So EFAM is under Interpro, ProSci, and many more. So you can look at protein families, classified protein families, protein domains, and others as well. You have Uniprot knowledge base that also houses a lot of functional information on proteins. Okay. Ensemble. So Ensemble has genome sequences of many model organisms and its annotation. So you don't have to manually download human genome and start annotating all over again. Annotations are already available. So if you are working on, let's say, cancer project and you want to look at specific genes, then you just need to go to Ensemble. They have a track for Homo sapiens. You can get the entire human genome which has been annotated and manually curated. Right, so the annotations are very accurate. And so DraftSeq is another example of secondary database. We also have disease databases. They, for instance, have COVID-19 database as well, HIV database, many disease-based databases are available. So these are all specialized databases. So it can be species-specific, disease-specific, or of a particular research interest. So example, flybase. So that houses the genome of the model organism. They also have HIV sequence database. So all HIV genomes that have been sequenced are available in this database. And there's also ribosomal database project. So all ribosomal sequences of bacteria and fungus can be found in this database. So these are databases that specialize in either a particular organism or a particular type of data, disease, or variation or mutations. Okay. Now we'll go more in detail into genome databases. This is what you'll be using for your project. So primarily we will be using DDBRC. So next Monday I will be introducing you to this database. Okay. So it's a searchable database which has bacterial and viral genomes with visualization and analysis tools as well. Then there's also other platforms for doing comparative genomics. So this one, COGD, it's quite a useful analysis tool or a software suite which interlinks and interconnects web-based tools where you can visualize, you can compare multiple genomes, you can see similarities, dissimilarities, missing genes or new genes that are found in one genome and not in the other. These kind of things you can visually observe using this platform. Right. And many more, ensemble genomes, human genome variation society, for instance, the last one. This is a society which has been formed to house all the 1,000 genome projects around the world. Have you heard of this 1,000 genomes project? So many countries have actually done this, and some have just recently started, where they have a pool of money to sequence 1,000 of their own citizens. Okay, so the idea is to get a reference genome of their own kind. So let's say Indonesia, they have also started this project in Malaysia, but maybe I'll give you another example. UK, for instance, that was one of the first countries that started this 1,000 genomes project. So they picked breeds, okay, that are classical Caucasians. That means they must have had ancestors from UK. So they pick them randomly and sequence their genome. So the idea is to have one reference genome which is very British. Okay. So for instance, that's the idea. So every country is now trying to make one reference genome of their kind. Because each of us, of course, our genome is similar, we are Homo sapiens, but we have our own variations, right? Mutations. So for instance, me and her, we look very different, although we are both humans, Malaysians, but one is Indian, one is Chinese, right? So we have variants or variations in our genome that makes us different. So this is what they want to try to come up with, like a reference genome. So for Malaysia, it's not going to be easy because we have multiple races. So I think they started off with having a Malay genome because it's like our majority population of Malays. So they have now built a Malay genome, but they are now trying to do 1,000 genome of Malaysians as well. So I think they're still selecting their participants. I'm not sure what the criteria is, but eventually we will have a reference genome for Malaysia as well. So for every country, they're trying to do this. So those countries that have already done this 1,000 genome project, this is there is a database where they actually submit the data to. So in future, if you are keen on studying a specific population of people, let's say you're interested in studying Malaysian Malays and certain disease that is related to that, or Malaysian Aborigines and diseases related to them, you can find genomes of that population in this database, right? So that's the whole idea, and this particular database, they are maintaining this data for this purpose. So in future, if people are looking at studying genetic diseases as very, very specific to certain population, these genomes will be very useful. Right, and many more. As I said, there's flybase, the Saccharomyces genome database. So that has the model organism, the yeast. The genome is there. The annotations are always updated. So if there is a new update, new annotation, they are updated in that database. Then there's worm-based zebrafish information network. So that houses the genome of zebrafish and its annotation. And then if you're keen on malaria, for instance, tropical disease, then there's also UCSC malaria genome browser. So you can actually pull out specific serotypes of malaria and look at annotations, look at what genes, what proteins are there, right? And many more. And I said this one particular platform is very suitable for your comparative genomics project data. So this is like a software that helps you do comparisons, visualization, to help understand evolution and dynamics of genomes in the future. Right? And it's free, so you can anytime go to the website and look at what they have. Then there's also UCSC genome browser. So this includes a broad collection of vertebrates and model organism assemblies. So you can actually select, let's say you're interested in studying human genome, you can select human genome, and the entire, you can visualize the genome in the sense you can see genes, proteins, specific mutations are all pulled out as well. And they used to have something called UCSC microbial genome browser, where they had all the bacterial and archaea genomes available. But now they have consolidated it to call it Genome Archive or GeneR. So this has collection of all the microbial genomes, and you can do visualizations here as well. If you search for GeneR, they have a link, and let's say you are interested in E. coli, you can select the name of the bacteria. When you click it, you will actually, it opens a page where it shows the genes. You can scroll the entire genome to see end-to-end how many genes are there, what proteins, the length, size, everything. Okay? And then, of course, it's not just limited to nucleotide databases. There are RNA databases where you have RFAM, is a database of RNA families, and then they also have mirror-based, or it's a microRNA database. So if you are eventually venturing into something very specific, like specialized, if you're looking at just microRNA, they actually have microRNA database from 271 organisms. And many more. There's also protein sequence database. collection of protein families. They are represented by multiple sequence alignments and hidden markup modules. So if you have, if you're studying a specific bacteria and a specific gene, you want to see the function of the protein, whether the protein has a specific domain. You will normally go down to EFAM database to look at what the domain does, what's the structure like, and all that. PDB is similar to how your NCBI is. They also have a Japanese counterpart, they also have a European counterpart. So all three of these databases, Protein Interbank Europe, in Japan, and Research Collaboration for Structural Bioinformatics, they all house the same data in all three databases. So if you upload your data in, let's say, the European one, the same data is available in the other two databases. So here, the data they upload is NMR and X-ray NMR and X-ray crystallography data. So if you have already elucidated the structure of a protein, this is where you upload the data. Then there are also metabolic pathway and protein function database. So if you're keen on only looking at specific metabolism pathways, or like maybe you're studying a pathogen, a bacterial pathogen, and you want to see the pathogenesis, maybe certain pathway actually causes the bacteria to be pathogenic, then you may look into metabolic pathway. There are databases that has the information about the pathway, the genes related to it, and the proteins involved, proteins and enzymes involved in the metabolic pathway. There's also Reactome. It's a navigable map of human biological pathways. So it ranges from metabolic processes to even hormonal signalings and many more. You can see that a lot of these databases are curated, meaning manually people have checked this information so you don't get inaccurate information. When you have primary database, for instance NCBI, Gene Bank, people don't curate the data. Whatever you put is what you'll find. So no one sits and checks whether you really got the sample from this specific location, whether you have analyzed it properly, whether there was any problem with the sequencing. No one looks at that. But some of the secondary databases, like your pathway database and all that, they're curated. So someone actually sits behind the scene. When you upload this information, they check through your experimental data to see what you've uploaded is accurate or not. And then there's also Bio C1C database. So this includes multiple other collections. This has both curated and non-curated information. And many more. Okay, and CART now. This database is becoming more and more popular as people are looking at antimicrobial resistance studies a lot. So this database has a collection of resistance genes and the sequences of their products and associated phenotypes. So these resistance predictions are done for 263 pathogens of interest, and they are frequently updated. So if you're looking at studying antimicrobial resistance, CART is quite a popular database that's frequently updated. Then there are also metagenome and metatranscriptome databases and tools. For instance, if you're studying microbial or bacterial diversity, then there's 16S metagenome database, such as Silva and Lean genes. So you can use them to identify what bacteria is there in your samples. And then there's also shotgun metagenome databases, such as HOMD, which is Human Oral Microbiome Database. So in future, if you're doing project related to dental plaque or oral microbiome, then this is a database that will be very useful. So you can use a database to do comparison and comparative analysis to see what's there in your sample as compared to what's there in the database. And tools are basically any software that you use to analyze or interpret your data, right? So these are some of the tools that are useful for metagenome and metatranscriptome PIPR was for viruses, and then as you see as browser and EPRC. So next week, Monday, during our practical, we will be learning how to use EPRC, which will be used for your projects as well. Right? Now, this is an old tool. It's also used for comparative visualization, but it's no longer being managed or maintained, so we won't be studying this. We will use what's available now. Right? Any questions? So, no questions. Let's can you please sit in your group and come and we